This matters because these questions about origins are not strictly scientific. They never have been. They weren't for the ancient Greeks. They weren't for the people of Israel. They're not that way for us today. They weren't that way for Darwin. These questions of origins obviously involve a lot of science, but they take us into philosophy. They take us into theology. They take us into our moral behavior, how we deal with each other. These multiple dimensions make these topics both very interesting and very challenging.
Welcome to the Creation Today show, where we bring together interviews with experts and solid Bible teaching. Your host, Eric Hovind, affirms the ultimate authority of God's Word, the truth of creation, and why it matters to you.
What happens when a little worm doesn't fit the evolution narrative? Well, you either ignore it, rewrite the story, or if you're honest, admit the theory has some serious wormholes. Today on the Creation Today show, we're diving into the world of a tiny round worm. I mean, we're talking small, just 1,000 cells, but don't let its size fool you. This little creature is packed with complexity, precision, and design that screams purpose, not chance. Hey, if you're new to the Creation Today show, we are on a mission to disciple the world. We want to turn your stumbling blocks into stepping stones on your journey to know the truth. My name is Eric Hovind, the president of Creation Today, and joining me is Dr. Paul Nelson, a senior fellow at the Discovery Institute, which is the leading voice in the intelligent design movement. We are going to unpack why this tiny worm is a massive problem for the evolutionary theory. We're also going to explore the critical differences between intelligent design and young earth creation and why those distinctions matter for Christians trying to stand on the authority of God's Word. Dr. Nelson, welcome to the Creation Today show, sir.
It's great to be here.
You were featured in several films that I have absolutely fallen in love with. Expelled, No Intelligence Allowed back in 2008. Several from Illustra Media over the years. Unlocking the Mystery of Life, Darwin's Dilemma, Origin, Design, Chance, and the First Life on Earth. And then Is Genesis History? You have a PhD in the philosophy of biology and evolutionary theory from the University of Chicago. And I got to say, sir, you have spent a lot of time in this conversation. Why is this so important to you?
Well, I have to tell you, I never intended to do this when I was younger. I wanted to be a painter and filmmaker. And no, I'm serious. I actually went to art school for 2 years, and I was so unhappy. I finally said to God, all right, I'm miserable. You obviously don't want me to be a painter. What do you have for me? What should I do with my life? And God has a sense of humor. We're created in his image. And he said, well, how about philosophy of science and biology? Obviously, I'm compressing many months of struggle, into a soundbite, but it really wasn't what I wanted to do. But I found these topics, and it's a wonderful thing to know what you're supposed to do with your life. And I never looked back.
Wow.
My passion for biology has only grown deeper with each passing year. And this matters because these questions about origins are not strictly scientific. They never have been. They weren't for the ancient Greeks. They weren't for the people of Israel. They're not that way for us today. They weren't that way for Darwin. These questions of origins obviously involve a lot of science, but they take us into philosophy. They take us into theology. They take us into our moral behavior, how we deal with each other. So these multiple dimensions make these topics both very interesting and very challenging. But I've never looked back.
Well, it's amazing to me how far we've come, so to speak. And do you see the world of biology? I mean, I'm thinking when you started, what was this, maybe 30, 35 years ago?
Oh, I'm old. I get senior discounts now. Okay, I'm a grandfather.
Ah,
No, really, and I've been doing this conservatively. Conservative estimate would be almost 45 years. And
45 now.
45 years, can you believe it? And actually, the changes that I have seen in biology alone have been so dramatic. Whole genome sequencing, the ability to look at, as we're going to do in this hour, look at the development of small organisms in incredible detail. So really, I think biology as a science is right now in its golden age. The tools that we have to understand living things are so far beyond what I started out with, really, as a college kid, that I never quite know what to expect when I open up my email on any given morning. What new surprises are going to be out there?
That's kind of what I wonder is, okay, if I can stretch it by 5 years, you've got half a century almost to make it sound really bad of experience watching us learn more and more. And I go, what's the next half a century gonna unfold for us? So
Seriously, yeah, yeah.
it's exciting. Well, catch us up on this fascinating tiny worm that I'm afraid to even try to pronounce the scientific name of.
Yeah, don't bother pronouncing the genus name. Just call it C. elegans, and every biologist will know what you're talking about. So this little worm was picked up as an experimental subject in the early 1960s because a number of molecular biologists felt like the real puzzles in their field had been solved, and they wanted to understand the nervous system. Well, C. elegans has a very simple, relative to us, very simple nervous system, but they soon discovered that its cell diversity and the way that it developed were also interesting puzzles. So as an adult, the worm, which is only a millimeter long, you can see it if you've got a pretty powerful hand lens, but it's quite small, and they're abundant. You can go out in your backyard and dig around if you have a compost heap, and you will find this worm. They consume bacteria in their natural environment. uh, 1,000 cells in the adult, roughly, give or take a few. That's a relatively simple animal. Interestingly, they have about as many proteins as we do. Okay, that's humbling, right? They've got about 19,000 protein coding genes. We have estimates are, in the low 20,000s. So we're not that far
Whoa.
apart, right? Are you a man or a worm?
Yeah.
But what really attracted me to these creatures was an evolutionary puzzle that I think you could say preoccupied neo-Darwinian biologists in the 20th century. Here are five of them, five whose work I studied in depth, and their work sort of formed textbook evolutionary theory in the late 20th century. So I would have read and did read work by all five of these guys. But the puzzle that they never solved is, I guess you could say, amplified by C. elegans, by what we know in particular about how it develops. And it's unsolved today. It's unsolved in May 2025. It was passed on, in fact, like a baton to the next generation of evolutionary biologists. So this guy, John McDonald, who's a geneticist at Georgia Tech, was a student of Francisco Ayala, who was himself a student of Theodosius Dobzhansky at Columbia, one of the people I showed on the previous slide. And when I was an undergraduate, so we're talking the early 1980s, McDonald published a major review article where he said, the 20 previous years of research on the genetic basis of adaptation has led us to a great Darwinian paradox. Now, in the original, when you look up this article in the original, he puts this in italics. He doesn't want you to miss it, the paradox that he describes. So he's talking about his training, all right, his undergraduate, graduate, doctoral, postdoctoral training. That 20 years had led him to a great paradox. And the paradox arises from the intersection of three ways of thinking about biology. So it's the overlap in the Venn diagram that I'm about to show you. The first is what C. elegans showed biologists, how it develops. And you can watch this directly. You can watch it not just in C. elegans, but you can watch it in fruit flies and mice and frogs and other so-called model systems of developmental biology. So this is direct observation, this first category. Then we have the theory of natural selection. Uh, uh, Articulated originally by Darwin, of course, and then in much greater detail with mathematical formulations in the 20th century. And then lastly, we have the theory of common descent. Now, common descent we cannot observe. It is a hypothesis about the past of life on this planet. But when you bring these three approaches to biology together, there's a paradox that lives right in the center of that Venn diagram. And that's what I'm going to describe in the next 20 minutes or so. So are you tracking with me so far?
I'm tracking. I love that you just go ahead and admit, hey, the common descent idea is a hypothesis. That's an interpretation of the data. It's not the data. And I know that's kind of what you guys have harped on a lot at the Discovery Institute, to say, listen, this is a question right here. Okay.
This is a question, and in fact, it's something that I'm writing a book on right now. I did my PhD on this topic now almost 30 years ago, and it'll have to be a separate conversation. But there's a lot of genetic data that are, I would argue, impossible to fit into the common descent framework. But in any case, it is a hypothesis and it's testable. Under the right circumstances, it's testable. So let's look at the developmental part of the paradox first. We'll take each of these circles and then we'll look at what happens when they overlap. So Drosophila is probably the best studied model system in developmental biology. And I think pretty much any biologist would agree with that. It's for over 100 years, it's been mutated in just about every way imaginable. So we've got a lot of knowledge about these little fruit flies. Then we've got the purple sea urchin. Not as widely studied, but still very well understood in terms of its genetics and development. We've got C. elegans, who is our star performer today. The zebrafish, Danio, which is very well studied, and mice. Mice are the standard model system for mammalian development, for a whole variety of reasons. Now, if you look at the length of time that these groups have been studied, as I said, Drosophila, we're looking at almost 125 years now of intensive research, going back to the beginning of the 20th century. The purple sea urchin has been studied really since the Civil War. The point is, each of these model systems has a very large research community associated with it and a very, very large literature, scientific literature. So these are not peripheral players in biological study. They are very well examined. And they are complicated. They're complicated systems. So C. elegans is the simplest, about 1,000 cells in the adult. But if you look at these other systems, what I'm going to describe for C. elegans will apply even more so to them. So there's a philosophical term, one of those things that gives philosophers a bad name. One of those phrases, a fortiori, which means even more so, right? That's a Latin phrase meaning even more so for these other systems. So if you look at them, mice, a whole lot more than 1,000 cells. There are 10 to the 5 just neurons in the zebrafish. A lot of cells in that little fruit fly. The purple sea urchin, which in its adult form sits in the water column on the substrate, has to deal with a lot of creatures that would like to eat it, like bacteria in seawater. So it's got a very sophisticated immune system. My point is, the puzzle that I'm going to describe for these worms applies even more so for the other 4 systems that we're not going to look at. Very simple thought experiment here, and I'll see if you guys can answer it correctly. What is the most important component in any cell phone? I've got mine right here, iPhone 15. Think about this now.
I'm on the 14. Boy, the most important. I guess off the top of my head, I'm thinking, well, how do you define that? Oh gosh. Okay. All
I'll
right.
cut to
All
the
right. You
chase.
got me.
I'll cut to the chase, right? If you don't, I mean, I'm dutiful about this. I don't like it getting below 75. My wife and my daughters will let it go to zero all the
Oh, I
time.
can't stand that.
I can't understand it, right?
Uh,
If you don't plug this in, on a regular basis, you get a very expensive paperweight.
that's right.
So here's a little sort of responsibility flow chart. If you look at these other systems, the CPU, the camera, all of the other systems, they all depend on having power. So
Wow.
the power supply, in terms of the global set of functions that an iPhone or any smartphone needs to perform, flows back to the battery. And the battery flows back to actually the electrical grid here in Northern Illinois, where I live, right? So the same logic applies to animal development. Not every event in animal development is going to be equally important. So
Hmm.
most animals, not all, most animals begin with a fertilized egg, the zygote, right? That's how we started our existence. That's how these worms start their existence. A single cell. The egg is fertilized by the sperm, and then what you have in the case of C. elegans is an unfolding tree of developmental decisions from one cell to approximately 1,000 in the adult worm. If you think about this, you've got a geometric increase in cell number 1, 2, 4, 8, 16, 32, and so forth, which is about 10 rounds of cell division from the fertilized egg to any cell in the adult worm. But if you look at that cone or that expanding triangle of decisions, it should be intuitively obvious to you that those early cell divisions are going to carry a much more, I guess you could call it, causal responsibility than events that occur later, just because of the geometry of how development works. And what happens in development, two things mainly: cell number goes up, but tissue specification and cell differentiation also increase. So as that cone of decisions is expanding, cells are being given particular jobs to do, which you can see when you look at the anatomical diagram there. Some of them are going to be nerves. Others will be in the gut. Others will be reproductive structures. Others will be muscles and so forth. And there's a logic here. You can't just double cell number. I mean, you can, but what you will get is what biologists call a shmoo, right? It's just a mass of cells. And you do not want in your own biology, none of us do, uncontrolled proliferation of cells, right?
That's called cancer, right?
That's called cancer. You've got it.
Yeah.
Uh, you want all the cells in your body to stay with their original job description, right? And don't go rogue on me. Don't, metastasize and just proliferate. So there has to be precise cell movement and directional vectors. This is the case for any developing animal that will give you a worm or a mouse or a fruit fly or a human being. Those things need to be controlled, and they're controlled by a logic. So I won't read this whole thing. I'll summarize it. Bruce Wallace was a leading neo-Darwinian biologist in the 20th century. He was a student of Dobzhansky at Columbia. And he said, you can think about the body plan. This word here, Bauplan, is the German noun for body plan. The body plan of an organism is set in place by a series of genetic switches. They control the logic of development. So you can look at it this way schematically. You've got a logic of developmental switches starting with that single cell that lead to the adult. And again, if you look at the structure of that unfolding tree of decisions, it should be clear to you that causal responsibility goes up as you go back in time in development, such that if you have a lower causal responsibility closer to the adult for any particular cell division, it's going to be correspondingly higher as you go back. It just falls right out of the logic of how it works. And what happens then is random changes in that developmental logic become increasingly destructive the earlier they occur. So what Wallace points out in this article, the very next paragraph, is he says the problem with changing the body plan of an organism is you have to change the logic of development in a way that still allows the creature to be functional. In a talk I gave, actually, it's available at YouTube about butterfly development, I point out that no human parent wants to be in the obstetrics suite waiting for the child to be born and have the doctor come in and tell the parent, your child has a macromutation. That's chilling to me. I mean, I've got two daughters, both of them blessed with good health, but I can readily imagine in the hospital in Boston where they were born, the terrible tragedy of having the OB-GYN tell my wife and me, your daughter has a macromutation. We know what the consequences of these changes are. They tend overwhelmingly to be destructive or lethal. So if you look at the logic tree here, a mutation expressed early in development may affect hundreds, thousands, or hundreds of thousands of cells downstream. Again, because of the logic of development, whereas something that occurred later may not have so many negative consequences. So this may look like a scary diagram. It's not really. What this is won the Nobel Prize for John Sulston, who actually sat at a microscope with special lenses, special microscopy in England for month after month, and he watched worms develop. And
Wow.
he traced out this complete architecture of development. So up here at the top, that's the single cell, the fertilized egg, and it immediately divides to two daughters. They're named AB and P1. And then those daughters divide, and you have this unfolding tree. It looks like a wiring diagram, I guess you could say.
Yeah.
So each of the terminals along those lineages will have a specific role to play in the adult worm. So the AB lineage gives rise to neurons, muscle, and hypodermis. The MS lineage also gives rise to muscles, but to particular glands, neurons, and cells called coelomocytes. Over here, I don't know if you can see it. It's you guys are in the way for me, but there's another group over here. They're germline, the cells that will give rise to the next generation. They're immediately isolated. They're kept off there to the right. And then I want to focus on this group, the cells in the E lineage that give rise to the gut. But you can see the logic here of how this unfolds. Yeah. And it's definitely worth a Nobel Prize to sit there and watch these worms develop month after month. So let's focus just on these cells, these 20 cells right here. They're in the gut. And you can see the level of detail that was necessary to trace these lineages out. So we're going to focus just on these 20 cells along this long tubular intestine. And here is the E lineage. Now, the E lineage is already 3 cell divisions down from that single-celled starting point. So we've already gone through 3 rounds of division. Now, what E will do is it will divide like this, and its daughters will divide, and then you get to those 20 cells. Now, if you look at the logic of this, you realize if you see that little red triangle there, that cell division is only responsible for these 2. Okay, it's kind of like a marching band, right? Like you're a tuba player, and your only responsibility is to make sure that the trombone next to you ends up next to you when you spell out, Auburn on the field. That's your only job. You lead yourself and the trombone out to the hash mark, and then you're playing whatever the fight song. But as you go back up the lineage towards E, the causal responsibility of each cell division goes up. So that division is responsible for 2. This one right here is responsible for 5. These 5 cells. So the instructions for those 5 cells in their terminal role in the gut are carried right here. Right? So this cell's got more responsibility. It's got to worry about all 5 of these. This cell division, has these 10 cells to worry about. So you can see as you go back up the lineage, the amount of information that has to be preloaded to get these cells in the right position is going up correspondingly. And up here, 20. E has to have the instructions for all 20 downstream cells, which means it's got to be front-loaded with more information. Now, take this logic and go back up to that single-celled starting point, and you realize just how much information has to be present in that single cell to get the whole lineage to unfold correctly. So I did this just because you can in PowerPoint. I scaled mathematically the level of responsibility for E compared to any of its downstream children, if you will. It's got a much bigger responsibility. So there's a technical term in developmental biology now that's due to my PhD advisor at Chicago, Bill Wimsatt, wonderful guy who developed a whole theory about this of what he calls generative entrenchment. So the argument would be E is deeply generatively entrenched relative to any of its downstream terminal cells. So you can do this for the major lineages that come down from the zygote, A, B, MS, E, and so forth. AB carries a very heavy responsibility, but all of these have some downstream consequences to changing the information in them. Increasing degrees of generative entrenchment. Now, why is this a problem? And I'm going to try to finish in 5 minutes. Okay, I think we're going to make it.
Oh, you're all good. This is fascinating, by the way. This is, I think I'm seeing where the problem's going, so I'm really, really interested in this.
All right, so let's put the next circle in. Natural selection has 3 necessary and sufficient conditions. You've got to have variation in traits. So I have brown eyes. I'm 6'1", roughly. My wife is actually taller than me. She's 6'2.5". She literally looks down on me. I mean, she loves me, but she does look down on me. Those variations have to make a difference to your reproductive output, and they have to be heritable. So these are like 3 legs of a stool. If they're all present, natural selection will occur. So I'm not denying the reality of natural selection. What I'm saying is, what can natural selection do? And in a case of development, the problem is these deeply entrenched developmental decisions, if you will, do not like to be changed. We know that very clearly from fruit flies, from worms, from mice, and so forth. The problem is, as I pointed out earlier, changing these early developmental decisions cascades downstream. And basically, the organism that's developing does not know where to go because so many downstream instructions are affected by these random changes. They're catastrophic. And selection eliminates them. There are no stable populations of fruit flies with no head. Or, right? They're gone. You might get that mutation, but it's so destructive, natural selection just sweeps it out of the population. And this was well known early in the 20th century. So Ronald Fisher, one of the founders of neo-Darwinism, said it's definitely pathological or deleterious or lethal in the wild state. All right?
Wow.
So the kinds of variations you need, you don't see. And even if they occur, they're not heritable. So natural selection can't work. Let's put the last circle in. Natural selection can't work for this level of change.
Right.
All right.
It's because what we're seeing it does not happen. It doesn't create new things. It's not, well, it's deleterious most of the time, correct? The mutations are bad.
They're bad, and there's no mystical or hard to understand reason. It's just a consequence of the logic of how developing animals are built. Now let's put it in the last piece, and we'll be done with this first part. Here's a standard family tree for the animals. Now, one thing the viewer should realize is all the actual animals are up there at the top. We've got pictures of them, right? Down here, these are conceptual structures. They're abstractions. And this one, for instance, this node, bilateria, just means a bilaterally symmetrical animal. But there is no actual organism called bilateria. It had to be there if this happened. Something like that had to be there. But you need to keep in mind, only the tips are the actual creatures. Everything below that in the branching diagram, those are, I guess you could call them systematic abstractions. They're created by the assumption of common ancestry, but they don't actually have corresponding evidence, really at all. All right. So you still, though, have to describe them if they existed. So here is a hypothetical bilaterian, an early animal that would have given rise to all the bilaterally symmetrical different body plans. So this was from a Russian zoologist. And you'll notice it has some features. He calls it urbilateria, that's original bilaterian. It has obviously bilaterally symmetrical morphology. It probably possessed a single cell stage during its reproductive cycle, that is, an egg that's fertilized by sperm, and it would have undergone a developmental process in each generation. So these are consequences of taking the animals we actually know and abstracting back in time and saying what features would need to be present in their original bilaterally symmetrical ancestor. Now, the same problem would arise that we just described for C. elegans. Even if urbilateria had, let's say, 5,000 cells, very, very small animal, and it's going through a developmental process from a single-celled stage, the same mechanistic difficulties would arise for its development. You can't escape them. And again, it comes directly from the logic of how development works. So there's no reason to think, on the basis of what we actually know about how animals develop, that urbilateria could have escaped this problem. All right, I made it. Slide 29. We're here, and in the first 30 minutes. So Charles Marshall, the gentleman here on the left at the bottom, was someone I interacted with briefly during my time at University of Chicago. He was there when I was there. And early in his career, he's an evolutionary paleontologist. He and one of his mentors in Australia said, we've got a problem with macroevolution. There must have been some way before the animal groups originated that their genomes could have changed dramatically without these downstream negative consequences. So as they put it in this publication, primitive genomes, early animal genomes, must be able to change rapidly in coordinated ways that are viable. In other words, they did something that we have no reason to believe today on the basis of what we know about developing animals. But if common descent of the animals is true, if that tree of relationships is true, something like this must have happened. The problem is the existential quantifier here, some, all right, gives away the game because they're saying there's some unusual feature. Who knows what it is, right? So in the second half, I will describe the problems with this hypothesis in relation to how we normally test things in science, or frankly, in daily life.
It seems to me like all of this, the bilateral, the imaginative, is all developed in an attempt to prove something. It seems like the starting point, where they're starting from, is giving rise to these concepts and ideas that aren't working, but they keep trying to create a way for the theory, the starting point, to work. That seems to be the unmovable idea that they can't seem to get off of. And I want to ask you about that and why that is, as well as the whole, you're in the intelligent design movement and you believe biblical creation. How do we think through this? So I got more to talk to you about. Hey, social media and television audience and podcast listeners, I need to let you go now. I'm so sorry. I would love for you to join the rest of this conversation over at creationtoday.org. Just come on over and join this conversation. Let me tell you where I know this is going to end up going. It's going to end up going to the place that a lot of us have been saying for a long time. You've got some serious problems when it comes to evolution. And this tiny worm is a big problem for the evolution worldview. If evolution isn't true, there's only one other game in town, if you will, one other idea, and that is the whole world and the entire animal kingdom and all of humanity was created by God. And he created you for a reason and for a purpose. And I'd love for you to discover that purpose. Mark Spence and I wrote a nice article for you to understand what that is. And you can have it for free at creationtoday.org/gift. creationtoday.org/gift. I really want you to understand who you are, why you were created, and how to have a relationship with your creator. If you want to get in touch with Dr. Nelson, what's the best way for people to get in touch with you? Is it through your personal social media or right through the Discovery Center?
I'm sorry. Just go to Discovery Institute and there's a site there where you can click and say, put me in touch with Paul Nelson, and they'll put you in touch with me. So just go to discoveryinstitute.org.
And you're going to want to read his works there. His articles are there. Lots of great information there. Really appreciate it. All right, looking forward to seeing you guys next week. I got a great show for you. Until next week, go share the gospel with somebody who needs to hear it. God bless.